Hello everyone, I am Shaili Dash. Welcome to the Knowledge Club online. In previous lecture, I discussed on types of immune system in our body and about innate immune system. We know now our immune system is two types. Innate immune system, which called natural immunity, and acquired immune system or adaptive immunity. Today, I will discuss on acquired immune system in easy manner. Adaptive immunity occurs after exposure to a foreign agent, improves upon repeated exposure and is specific. It is mediated by antibody produced by B lymphocytes and by two types of T lymphocytes, namely helper T cells and cytotoxic T cells. Details on these lymphocytes we will discuss in another lecture. I will discuss today a general overview of acquired immunity system. So let's start discussion. Before that, if you are first time in our channel, please subscribe this channel and activate the bell button by clicking it for getting our every video update. And if you subscribed already, then thanks a lot for your supporting and follow our videos regularly. The innate immune system is required to ignite the adaptive immune system. Acquired immunity is of two types, active and passive. Now, what is the active immunity? Active immunity is the resistance induced in and follows individual after effective contact with an antigen. It follows either natural infection or vaccination. Here, the immune system actively participates, producing antibody and often cell-mediated immunity also. Active immunity develops slowly over a long time, like days or weeks, but persists for a long time, usually for a year. Active immunity may be acquired either natural or artificial. Means it's two types, natural active immunity or artificial active immunity. In natural active immunity, it is acquired by natural infection by the organisms, predominantly by the subclinical infections after repeated exposure to a small doses of the infecting organisms which passed unnoticed. Such immunity is usually long-lasting and plays important roles in preventing epidemics. Example like poliomyelitis, tuberculosis, etc. Some of these follow overt infection. Example like smallpox. In artificial active immunity, it is the resistance produced by vaccination. The vaccines are preparations of live, attenuated or killed microorganisms, or either antigens or active material derived from them, like toxoids. Some common vaccines are in bactericidal action, the live, BCG, anthrax, plague and Pusoli vaccines. In killed condition, TAR for enteric fever, auto vaccines, etc. In viral vaccines, live condition smallpox, measles, influenza, mumps and sabin. And killed condition, soft vaccines for poliomyelitis. Number 3, the bactericidal products, toxoids for tetanus and diphtheria. I used some terms in this case like attenuated, killed vaccines and toxoids. What are those? Attenuation of live vaccine is done by aging of culture. Cultivation at high temperature like anthrax bacilli grown at the 42 degrees centigrade. Passage through animal of defined species like rabbits and calf. Drying rabbits by continued cultivation in the presence of antagonistic substances like BCG and by repeated subculture in artificial media like Streptococcus. In killed vaccines, the organisms are killed by heat, formalin, phenol, alcohol, ultraviolet light and photodynamic inactivation. These are the preserved in phenol, alcohol, and martinolate. Then toxoids are preparation of bacterial exotoxins inactivated by formalin like formol toxoids or by alum, APT. They retain immunogenicity but lose toxigenicity. Now, mechanism of action in general of active immunity. The body's immune apparatus offers protection by synthesizing antibody by B lymphocytes and plasma cells and or by sensitized T cells in response to an antigen during the first encounter between the host and microbes. Antibody formation starts only after a latent period which is attributable to antigen combining with any pre-existing antibody in blood and lowering blood concentration level of antigen. Once developed, the active immunity lasts longer. This is called primary immune response. When an individual encounters the same antigen subsequently, the immune response is more rapid and stronger. This is called secondary immune system. Acquired active immune response takes two forms, humoral and cell-mediated, and both usually developed in parallel. Number 1, humoral or antibody-mediated immunity means AMI, and number 2, cell-mediated immunity CMI. So, in this humoral or antibody-mediated immunity AMI, there is production of specific circulating antibodies in the host which combine specifically with the corresponding antigens and modify their activity. Thus, antigen molecules or particles may be clumped or lysed. Their toxins may be neutralized and readily removed after phagocytosis by neutrophils or macrophages. Then, what is the action of cell-mediated immunity means CMI? It mainly depends on the specifically developed T cells by certain antigens. These sensitized T lymphocytes are important in resistance to chronic bacterial infections characterized by intracellular parasitism. Example like tuberculosis, brucellosis, leprosy, and also in some viral infections, that is herpes simplex. So this is the overall the active immunity. Now come to the point of the passive immunity. The resistance that is induced in the recipient by transfer of performed ready-made form antibodies against effective agent or toxin in another host is called passive immunity. The immune system plays no active role and the protective mechanism comes into force immediately after transfer of antibodies, the immune serum. Passive protection is short-lasting only for days or weeks. It is useful when instant immunity is required. Passive immunity is of two types, natural and artificial. Natural passive immunity is the resistance passively transferred from the mother to fetus and infant. The example, the transfer of maternal antibody to fetus transplacentally and to infant through milk means cholesterol. In artificial passive immunity, it is the resistance passively transferred to a recipient by the parenteral administration of antibodies. The passive administration of antibodies is very useful in some clinical conditions. After the first injection of foreign serum antibody, that is the ATS immune horse serum, it is eliminated only by the metabolic breakdown. But on subsequent administration of horse serum, it is eliminated much rapidly because it combined with antibodies to horse serum that has been produced following first injection. This is called immune elimination, which limits the usefulness repeated passive immunization. Adaptive immunity is a special type of antibody-related passive immunity related to B lymphocytes. If such cells or bone marrow is successfully transplanted into a B cell deficient individuals, the recipient will produce antibodies which previously could not be produced. The adaptive immune system uses a broad range of molecules for its activities. Some of these molecules are also used by the innate immune system. The broad range of molecules for activities of the innate immune system and the adaptive immune system I will discuss on the next videos. Today's lecture up to this. I am coming with the next video very shortly. Till then, goodbye.
So, if you like our video, please click at the like button and share this video in your circle. Don't forget to subscribe our channel, which will help you to gather more knowledges in future. And if you already subscribed our channel, then click at above bell button to know our channel updates. At last, please write your experience regarding this video at comment box, which will help us to evaluate ourselves. We are coming with our next video shortly. Till then, goodbye.